In this video, I will be talking a bit about aquaporins and the regulation of water and osmotic pressure inside human cells. And so I guess the first question would be, why would we need aquaporins, which are a type of transmembrane protein, to regulate water? Couldn't the water just sort of flow in and out of the cell on its own? And the reason that we want this is so that the cells can have some kind of control over the water. And so I have here, this is just a little crash course on osmosis. And so osmosis is if you have something that looks like this container here, where there's water on this side and then water with some kind of solute on this side. The water will flow into the side with the solute. And sort of the reason for that is because on this semi-permeable membrane, on the side without the solute, the water is going to be sort of, running into these permeable areas more often because the water is sort of more highly concentrated there. Whereas on the solute side, these solutes will, I guess you could think about it as like they sort of block these little entrances here, these little pores here, and so it prevents water from sort of flowing back in the other direction. So you end up with this osmotic pressure, and so you can actually see that the water is lower there than right there. And so this can happen with cells too, where if you are in an isotonic solution here, which means that the sort of solute content inside and outside the cell is relatively the same. And so that would be sort of, what the cells would want. And that's why, for instance, if you get like an IV drip, they use saline solution, which has a little bit of salt in it because they don't want to just put pure water into you, because then you could end up with, well, basically this right here, this hypotonic solution where the water is going to sort of osmose into the cells and cause them to bulge and then burst like this. They can what's called lysis, or lyse the cells, or burst. And so if you have too much salt, like in your, I guess, veins, these are red blood cells in this example, so we can think about it in veins. If you have like too high of a salt concentration, you have what's called a hypertonic solution, and then the water will actually sort of leach out of the cells, and so that'll cause the cells to shrivel. And so that is what osmosis is and how it relates to cells, and that's why we want these aquaporins to sort of help regulate the water content of the cell. You want these aquaporins to be able to sort of open and close depending on the sort of water needs of the cell. And so this here just gives us an idea of, all the different tissues in the human where we can find aquaporins. And there are 13 aquaporins. Humans have 13, so there's aquaporin we can call AQP, and it starts at 0 and then goes to AQP12. And so there are 13 of them, and they're sort of split up into 3 different groups. I think there's like the orthodox group and the unorthodox group, and I'm not going to really go into that, but it just, sort of the relatedness of the different types of aquaporins. But we can see here, we find these in pretty much every kind of tissue you can think of in the human body, and that makes sense because you want this water regulation in every part of the human body. Of course, we're going to be kind of focusing in here on how it relates to the digestive system here. I mean, I'm going to be sort of talking about aquaporins in general here, and just know that this is also the same mechanisms that apply in the digestive system. It seems like a lot of what I find, a lot of papers and information I find on aquaporins has to do with like the kidneys and the brain, as a matter of fact. And so, yeah, there's, papers on all these different aquaporins, but the thing to remember is that it's sort of the same mechanism in all of these. And so, even though some of the things we'll be looking at are from, other tissues, or the paper that I got it from was focusing on other tissues, the mechanism is still the same as in the digestive tract. All right, and so the aquaporin has this sort of topology here, where it has these six transmembrane domains. Then, you can see between the prokaryotic and archaeal, and then the eukaryotic, there's different sizes of these different loops here. And so we have this NPA here as well, which is this NPA motif, the asparagine, proline, alanine motif. And so let's actually look at that a little bit here. So this little picture here, which I got from Socratic, is just kind of showing sort of a schematized look at this of water sort of going through this channel. And what it shows here that's kind of interesting is that the water goes through one molecule at a time, and so it's actually sort of going single file. And I think the Socratic actually said that about 1 billion water molecules go through per second, and so that's a lot of water molecules going through every second through one of these things. And so this is actually a crystal structure of it, and you can see these little dots here go around where this this channel actually is. And if we zoom in, you can actually see these red spheres here are water molecules, and you can see those in the channel there. And these channels have this conserved aromatic arginine motif. So this arginine right here, then aromatic would be this phenylalanine here. And then, of course, we have this NPA motif, which is in yellow on here. So we have this this proline here. We have this asparagine here. We have this proline and this asparagine here. And so you can see that this has a lot of asparagines and prolines and alanines in this motif. And so that is actually, so if we look at this here, this is sort of looking again at this channel. And so we have the arginine, asparagine, and asparagine. Here's the NPA and this aromatic arginine motif here, which has this electrostatic repulsion. So you can kind of see these little red and white V shapes are the water molecules, where the red is the oxygen, the white are the hydrogens. So we see that this has a size restriction here, and this also has a dipole inversion, where we see coming in, the oxygens are kind of pointing downward, whereas when it comes out here, the oxygens are sort of pointing upward. And so there's actually, it's sort of flipping the water molecules over as they go through. And so these aquaporins, so water is sort of the main thing that they allow through, but I found in some other papers that some of these, some of these different ones, we have all these 13 different ones, actually allow things like ammonia and hydrogen peroxide and carbon dioxide and oxygen and things like that through. And in fact, I don't think I have that on here, but these pores actually form a homotetramer, and there's actually a pore so the pore that water goes through is inside this channel in the protein, but there's actually a pore that shows up between the 4 different monomers, and that's sort of proposed to be where those different gases like like carbon dioxide and oxygens and things like that can go through. All right, so here, once again, these dots are looking at the pore, so the path of the pore. And these are just looking at, I believe this SOPIP2 is a plant kind. And then these other ones, I think HS is Homo sapiens aquaporin 4. I think some of these other two are animal ones. But the thing to notice, and they kind of have it kind of boxed here, is these areas here where you can actually get this restriction of these residues into the pore, and those are controlling whether water can go through them or not. So these pores can all be closed if you don't want water moving through them, or they can be opened back up. And so this is looking at some of these residues here. So if we're looking at this tyrosine here, that's what we have down here. So we have this tyrosine and this tyrosine. And there's two of them here, and this is something you see a lot of in structural biochemistry. It's that thing where, like, if you're trained, you can sort of like look at it and see a 3D image, because these two images are sort of a little bit rotated from each other, and if you're trained, you can get that sort of illusion of seeing it in 3 dimensions. I have never been able to do it. I usually just end up getting a headache when I try, but that is, you see this a lot in structural biochemistry papers. But these are two just pretty much the same image, just slightly rotated from each other. But yeah, you see down here, this is where those tyrosine residues are, which can close that pore. And so this down here is looking at a few of these other different things. So for instance, this histidine here, which can sort of switch over and sort of get in the way of the pore here so that water can't make it through. Over here, this histidine, well, this is the same histidine as this one, so that can move over and get in the way of the pore. Same with this tyrosine, so this is that tyrosine 31 that we were looking at up here. And so what this is talking about is that these things are done through phosphorylations. So phosphorylations of serine 115, and there's also pH regulation. So like the histidine 193 is regulated by pH. And I actually have this table from a different paper here. And so this is actually looking at the different aquaporins and the phosphorylation sites on it, and the different proteins that can actually phosphorylate those sites. So we have like the protein kinase A, protein kinase C, protein kinase G, and things like that can phosphorylate these things. And this S is serine N, so Na, I guess is not applicable, but then T is threonine, and so those are those residues that have that hydroxyl group on them that can be phosphorylated. And then of course Y is tyrosine, which also has a hydroxyl group on it. And so these are all the places that these aquaporins can be phosphorylated, which adding a phosphate adds this negative charge to it, which can cause conformational changes in the protein, which could do something like this, for instance. Well, this one was actually pH controlled, and so histidine has a pKa that's fairly near physiological pH, and so that's often seen in pH controls. And so we see that if one of these nitrogens becomes protonated, then it could form this hydrogen bond here with this aspartate over here. So right here on this one, you can see that there's this red and this purple chain have this conformational change that occurs because of like a phosphorylation, for instance. And so there's sort of this internal kind of regulation using phosphorylation. But there is also the phosphorylation and ubiquitination that can actually be used for controlling sort of the expression of these proteins. And so here it says the phosphorylation is a ubiquitous mechanism in aquaporin regulation by both gating, which is what I've been talking about, gating, and trafficking. So we see gating here, we phosphorylate something and it could, maybe close this from allowing water to get through. Then you could dephosphorylate it with a phosphatase enzyme and open it back up and allow water to go through. But we also see here that we have these phosphorylations. So we have this storage vesicle here, and so this through phosphorylation would come up to the plasma membrane here and sort of merge with it. So it kind of gets split right here where this side joins that part right there and this side joins that part right there. And then it kind of puts this thing outside or pointing outside the cell. And then we see that there's this other phosphorylation that actually stabilizes it here. And then ubiquitination is, well, a rule of thumb is that ubiquitination is usually involved in sort of the breakdown of proteins. And so we see that if this gets ubiquitinated, then we would actually it would undergo endocytosis and get brought in like this. This vesicle will then go over here to this MVB, which the MVB stands for the multivesicular body. But then we can have this join onto the lysosome. And the lysosome contains enzymes that will actually break down proteins. So lysosomes are usually sort of acidic environments, but the acid, I guess, can break it down, but so are sort of acid activated enzymes. And, the reason you would want to have them activated in a different pHs so that, if they somehow sort of break out of here, then they would be in the sort of more neutral environment of the cell, and that would deactivate those enzymes so they wouldn't break down other proteins that you actually want to keep around. But anyway, so you can see that these aquaporins are, as I said, they are phosphorylated, which can do this gating mechanism, which is what we were talking about above, but also this trafficking mechanism, so bringing it in and out of these vesicles and ubiquitination, breaking it down. So that is how these aquaporins actually sort of regulate the movement of water in and out of cells. And as I said, the reason that we would want that to be the case is so that our cells can control the osmotic pressure inside of the cell better. And we want to be able to do that in pretty much every tissue in the body, and so that's why we see this. And so this is actually showing, because this is actually a paper that was talking about pathogens and how those actually affect the different aquaporins. And the pathogens can actually sort of release toxins that will either activate or inactivate these aquaporins in ways that our bodies wouldn't want them to, and that can actually cause some of the sort of pathology of different pathogens, bacteria and viruses and so forth. And the paper also talks a little bit about how there are some genetic diseases that are caused by, I think, particularly this trafficking mechanism here. There are sort of genetic disorders in the trafficking mechanism that can lead to, different sort of non-pathogenic sort of chronic diseases. But anyway, I hope you found this video useful, and in the next video I will start talking a bit more about the absorption in the intestine of different minerals. In particular, I'll be talking about like the sodium potassium pump, and I'll be talking about sort of absorption of calcium and magnesium and things like that. But anyway, I will see you in the next video.